Fourth and last among the chromosomal rearrangements, we have translocations. Unlike the other types of chromosomal rearrangements, translocations occur between different types of chromosomes that have different genes. Like inversions, translocations involve chromosome breakage, but instead of reattaching to the same chromosome, the broken region attaches to a chromosome of a different type, a non-homologous chromosome. So this is not crossing over, which takes place between homologous chromosomes. For example, regions 13 and 14 could break from this chromosome and move to between regions 4 and 5 of the pink chromosome. Since the orange chromosome receives nothing from the pink chromosome in return, this is called a non-recipricol translocation. More common are reciprocal translocations, where regions of chromosomes are exchanged between non-homologous chromosomes. For example, regions 13 and 14 might be exchanged for regions 5 and 6 of the pink chromosome. Like inversion, this does not involve any change in gene dosage. And like inversions, translocations can cause deletions. One way this can occur is if two acrocentric chromosomes both have small fragments break off and attach to each other, so that these fragments form a tiny chromosome and the other fragments form a large chromosome. Very small chromosomes can fail to segregate properly and eventually get destroyed. So the cell is left with a large chromosome that has genes from both chromosome types, but that is missing the genes that were on the two small fragments. This type of translocation is called a robertsonian translocation, and as we'll see in a later video, it can be responsible for a form of Down syndrome. Translocations can also create gene fusions. In fact, we've already seen an example of this when we discussed cancer genetics with a translocation between chromosomes 9 and 22, causing chronic myelogenous leukemia. Recall that if this particular translocation occurs, part of a gene from chromosome 9 is fused to part of a gene on chromosome 22, creating a fusion gene that encodes a fusion protein, which in turn can cause uncontrolled cell division. Although we didn't mention it on the last page, inversion can also potentially create gene fusions, since it also involves the movement of genes to different loci. It is also possible for a translocation to break a gene, which again would likely cause loss of function. This is probably more common than the production of a fusion protein, but could also promote cancer development if the broken gene is a tumor suppressor gene. Since translocations can move genes, these genes can exhibit position effects. We saw an example on the last page where a gene was moved near a euchromatic region, increasing its expression. Here's another example of a position effect. We see here just parts of two chromosomes. If a translocation moves genes from the yellow chromosome to near the centromere of the pink chromosome, the expression of those genes will likely go down, as centromeres are generally heterochromatic. If a gene is moved to near a highly expressed gene, the moved gene may also become highly expressed. We see this in the case of Burkitt lymphoma, a cancer of white blood cells, specifically B cells. It can be caused by a translocation between chromosome 8, which has the proto-oncogene c-MYC on it, and chromosome 14, which has the highly expressed IgH gene. A reciprocal translocation can bring the c-MYC gene near the highly expressed IgH gene, causing the c-MYC proto-oncogene to greatly increase its expression. Because this increased expression promotes uncontrolled cell division in cancer, the translocated c-MYC gene is now considered an oncogene. To summarize, we see that chromosomal translocation can have many of the same effects as inversion, including deletions, gene fusions, and position effects. Cancer often causes genome-wide instability, greatly increasing the rate of translocations and other chromosomal rearrangements. But as we've seen, rearrangements can also cause cancer. So it can be difficult to determine whether a rearrangement is the causal factor in a cancer or just a consequence of the genome-wide instability that is already present in cancer cells. Sometimes it's both, and of course it depends on the rearrangement and the type of cancer. That being said, researchers have, to varying degrees of certainty, found that rearrangements, translocations in this figure, cause or are associated with many cancers and other human disorders. We've already mentioned Burkitt's lymphoma and chronic myelogenous leukemia. Even some forms of schizophrenia and infertility are associated with translocations. This concludes our discussion on the 4 types of chromosomal rearrangements. Next, we'll take a look at aneuploidy. See you all then.